Hello, everybody. My name is Atsushi Tanaka, the director of St. Mother Hospital in Japan. Today's my title is How to Improve the Clinical Outcome of ROSI. Azoospermia is a condition that no sperm cannot be found in the ejaculate. The frequency is about 1 out of 100 healthy men. And azoospermia is classified into two categories. One is obstructive, 30% of all, but it has normal spermatogenesis, so normal sperms can be collected with MESA. The other is non-obstructive azoospermia, about 70% of all, and the sole treatment to collect sperm is mycotest. General success rate in Japan is about 30 to 40%, but about half of them are immotile or severely deformed. So, about 40% of non-obstructive azoospermia come to medical attention. This is a simplified diagram of spermatogenesis. Spermogonium develop to primary spermatocyte through mitosis and divide into two secondary spermatocytes. Each secondary spermatocyte divides into two round spermatocytes through first and second meiosis, and round spermatocyte transform to mature spermatozoon through spermiogenesis. As you know, yellow-colored blocked round spermatocyte and spermatozoa has the same number of chromosomes and the same content of DNA. So both of two gametes has the same capability to fertilize with oocyte. In 1992, Palermo developed ICSI. ICSI not only helped almost all patients with severe male factor infertility, but changed the concept of fertilization. So far, sperm-egg membrane receptor binding with interstellar calcium release is the mechanism of fertilization. But after the X cytosolic sperm factor plays very important role in fertilization. Round spermatocytes have a haploid set of chromosome and the same content of N, just as mature spermatozoa. So direct injection of round spermatocyte enable it to fertilize the oocyte. This is the rationale for round spermatocyte injection. In 1993, Ogura and Yanagimachi first reported round spermatocyte nuclei injected into hamster oocyte form polynuclei, which could participate in syngamy. And next year, they reported the first burst of PAP. In 1994, the late Professor Edwards, Nobel Prize winner, reported the idea of using spermatocyte as a substitute for spermatozoa. And in 1996, Tesalik reported the first successful lossy. And after that, many doctors reported the successful pregnancies, but actually very few reports of deliveries. Around 2000, many doctors became skeptical whether this technique is essential or not in clinical application. Then this ROSI gradually disappeared. But I'm confident this ROSI might be a great technique to save the patient is suffering from the male infertility. So we have been keeping the study and experiment. How should we interpret this phenomenon? When no spermatozoa or late-stage spermatozoa found in testes, but round spermatozoa were found, it has been accepted generally that whenever round spermatozoa exists in the human testes, there are also mature spermatozoa present. Frequently, I was asked many doctors, why Dr. Tanaka, do you do round ROSI? In the case, you could find mature spermatozoa with careful observation. However, the new facts recently reported. Mouse and human male with CRAM gene mutation showed the maturation arrest at round spermatozoa. And also TAF4B and ZMYND15 also show the same maturation arrest at the round spermatozoa. So I think it's necessary to reconsider the usefulness of ROSI. There are three big issues for extremely low success rate. First is difficulty in cytodifferentiation of round spermatozoa from other round cells. The most difficult cytological differentiation is between spermatogonia and round spermatozoa. I'd like to tell you how we had come to the goal. Almost perfect differentiation between two cells. We happened to find unfamiliar cells while we prepared the tissues of seminiferous tubers at microtesting. Spermatogonium is identified by its amoeba-like movement, pseudopodia. The small-sized spherical pseudopodia are emerging from the many places spontaneously and return to former shape in a minute. So I thought these round cells with pseudopodia might be spermatogonia. Spermatogonia are defined as the primordial germ cell, which have migrated into the testis with amoeba-like movement and ceased its movement when they reached at the seminiferous tuberous. However, we could observe a lot of round cells with this movement among the testes. So we started the experiment to prove our hypothesis is correct or not. This image is seminiferous tuberous of 10 days old mouse, very young. Seminiferous tuberous are consisted of several cells, spermatogonia, and a small number of primary spermatocytes. This slide shows frequency of round cells with pseudopodia. These cells were found over 50% in very young mouse. However, they decrease rapidly in adult mouse and about 2 to 6% in both of azoospermic and normospermic men. So we can list the cytological characteristics of spermatogonium, that is 8 to 10 micrometer diameter, a high LC ratio, 1 is 1 nor 2 prominent nucleoli close to the distinct nuclear membrane. This is a result of chromosomal analysis. Spermatogonia and the primary spermatocyte have a diploid set of 46 chromosomes. In primary spermatocyte, the crossover that are peculiar only to primary spermatocytes are found. When you could find the crossover, you can say these cells are during the meiosis. That is, this cell is 100% primary spermatocyte. Long spermatocyte has haploid set of 23. Following these various comparisons of spermogenic cells with cytological characteristics, fish analysis, and chromosome analysis, now we have come to differentiate these three kinds of spermatogenic cells almost 100%. A chromosomal vesicle or granule are found about 20 to 30% of maturation arrest of long spermatocyte seminous tuberous. And these findings are very helpful to identify it. I will show you the actual procedure of ROSI from now on. This is a view of the microdissection of the testicular cell extraction in the operation room. Thickened coiled seminous tuberous, which are likely to contain spermatozoa spermatocyte, are picked up under microscope. Biopsy tissue were macerated with a 27-gauge fine needle manually and a microdrop of medium containing DNase and collagenase to facilitate separation of individual cells. Specimens are observed under an inverted microscope equipped with the Nomalsky differential interference contrast system. Two spermatozoa could be found and cryopreserved for next step. When no sperm could be found, what should we do? It's Lossy's chance. Round spermatocyte, indicated by the red arrow, has a high NC ratio, 68 micrometer in diameter, which is one size smaller than spermatogonia. It has a clear nuclear membrane. Spermatogonia, indicated by the yellow arrow, has a high NC ratio, 8 to 10 micrometer in diameter, distinct nuclear membrane attached with 2 to 3 prominent nucleoli. Primary spermatocyte, indicated by green arrow, and cytosol by pink, are easily differentiated according to the cell size and chromatin pattern. It may be difficult to differentiate spermatocyte from spermatogonia because of their similar size. Unlike the nucleus and cytoplasm of somatic cells, those of spermatocyte and spermatogonia can be readily separated by sucking in and out of an injection pipette. Both types of cells are easily aspirated into pipette and separated into nucleus and cytoplasm. While the nucleus of round spermatocyte is homogeneous and clear, that of spermatogonia has 2 to 3 prominent nucleoli. Next is how to differentiate non-round spermatocyte. So non-round spermatocyte is very difficult to aspirate. And after repetition of aspirate and expel, the cell is not deformed and return to normal shape. And how to select the viable round spermatocyte. Viable round spermatocyte, the nucleus shows bouncy and a transparent pattern. In the case of non-viable round spermatocyte, the nucleus of non-viable shows distorted and atrophic nuclear membrane. Oocytes were transferred to a solution of mannitol with calcium and magnesium ions and exposed to an alternating current followed by a pulse of direct current. From here, I will show you the actual procedure of ROSI. The long spermagene was drawn into the injection pipette. Long spermagene was easily separated into nucleus and cytoplasm. Then both the nucleus and the separated cytoplasm were injected into the ooplasm. Now you can see the separated round spermagene is approaching the tip of the pipette and is inserted deeply inside the oocyte. You can see the injected round spermagene enclosed in green. It takes about 1 minute per one lossy. The technique is almost the same as ICSI. Next video is how to do piezo lossy. We have recently developed. First, we aspirate the round spermagene. Aspirated round spermagene was broken into nucleus and cytoplasm in the pipette. Piezo lossy was performed by applying a piezo pulse, which produced ultra-fast submicron forward momentum using flat-tipped micropipettes with no spike. Penetrated zone procedure was pushed out and the tip of the pipette was attached to the surface of cytoplasm and advanced forward to stretch the membrane. The breakage of the membrane was performed by applying one piezo pulse. The nucleus and cytoplasm were enclosed in green circle. The cytoplasmic damage was decreased by 30%, resulted in high embryonic development than conventional ROSI. Next is insufficient oocyte activation. At fertilization, repetitive calcium ion lysis called the calcium ion oscillation occur. This is due to the release of calcium ion from the endoplasmic reticulum through the IP3 receptor. Calcium ion oscillations are induced by sperm factor. A current strong candidate sperm factor is sperm-specific protein. It is phospholipase C-zeta. In the cytosolic aspiration and injection activation method, calcium oscillation were not occurred. You can see this video didn't show the expression of calcium oscillation. In the ionization activation method, fluorescent intensity gradually decreased after 5 minutes after treatment. You can see calcium oscillation could not be observed. This video showed calcium oscillation clearly after electrical stimulation. The first spike appeared about 15 minutes after the stimulation. However, height, interval, and duration of spikes were irregular. In two oocyte, you can observe calcium oscillation lysis when it becomes brighter and lowers when it becomes darker. This one showed calcium oscillation of electric stimulation plus ROSI. It induced consistent large peptide calcium oscillation. You can see in the image, you can observe the bright and dark changes alternatively, and in the rhythmic pattern. We reported 14 babies born after round-spine injection into human oocyte to PNAS in 2015. We selected only patients whose first microtest conducted by oncologists at other institutions showed no testicular sponsor or late-stage spermatocyte, but had round spermatocyte found at the second microtest performed at our hospital. Received ROSI in 204 treatment cycles. In this slide, you can find 9 babies were born in first cycle. Delivery rate was 5.8% and 5 from frozen cycle with 11.9% birth rate. However, high miscarriage rate is a big problem. This is a clinical data of parents of ROSI. You can find that 5 cases out of 14 show that Johnson's score was below 4 points. It means pathological results of all 14 successful cases has failed to develop to spermatocyte. We might say Johnson's score is not reliable to make a final diagnosis. We reported the result of follow-up study of 90 babies born out of ROSI for 2 years to fertility sterility in 2018. In conclusion, in this paper, we made it clear there was no significant differences of physical and cognitive ability between ROSI conception group and natural conception group. This figure shows the clinical outcome of ROSI from 2000 to 2017. We had more than 300 healthy babies until 2017. What should we do for loss improvement to decrease the rate of miscarriage? We reported the results of in vitro culture of round spermatocyte on the feeder layers of Vero cells to fertility sterility in 2003 to examine whether human round spermatocyte will differentiate in an in vitro culture with Vero cells. Isolated early round spermatocyte were co-cultured with Vero cells under various conditions. This figure shows isolated round spermatocyte on the monolayer of Vero cells. After 2 or 3 days, elongated spermatocyte with nuclear protrusion and condensation indicated by green arrow, and elongated spermatocyte could be found, and even fragile growth could be found, indicated by white arrow. This slide shows in vitro spermiogenesis of round spermatocyte obtained from non-obstructive men. We found about 30% of in vitro cultured round spermatocyte from non-obstructive azoospermic men developed to elongating spermatocyte. We had performed ROSI using these in vitro cultured cells, but clinical outcome had not increased, so we stopped it. However, after that, we have been thinking of the co-culture with cells to develop the round spermatocyte to further stage. One day, my friend, he is working for the IPS cell examination. He told me, Atoshi, cell to cells is very good culture cell. Why don't you try them? Because cell to cells has regulation of spermatogenesis and testis development, and the provision for spermatogenic cells with structural, immunological, and nutritional support. And these beneficial effects give stable global gene expression profiles and numerous proteins and the activation of AKT and SMAD 1 to 5. Long-term culture of human mature cell-to-cell have been regarded as impossible, but Guo et al. Reported a success case in 2015. We have been also experimenting the in vitro culture of cell-to-cells and was successful to establish the cell line. We are now planning to start the in vitro culture of round spermatocyte on the feeder layer of cell-to-cell. Last one is epigenetic errors. What is epigenetics? DNA modification that don't change the DNA sequence can affect gene activity. Epigenetic changes can help determine whether genes are turned on or off and can influence regulation of reproductive activity. There are two primary epigenetic mechanisms. One is DNA methylation, the other is modification of histone. Since histone platinum replacement is incomplete in round spermatocyte, it is thought that the programming of the paternal genome does not occur in the usual manner after lossy. The other one is active DNA demethylation. So, how to solve these problems? I am now thinking of two approaches. One is in vitro co-culture with cell-to-cell, and the other is histone deacetylase inhibitor, scriptide. My friend working for the ROSI way using mouse, he told me using something is necessary to improve the birth rate of lossy because birth rate of following lossy is 30%. On the other hand, X embryo success rate, birth rate is 60%. It's double. What's wrong? Why? Nuclear protein of round spermatocyte is histone, and elongated spermatocyte is a transition protein, and spermatozoa is pertanine. So these differences in three cells are causes of the big differences. Chen et al. Reported the effects of the histone deacetylase inhibitor scriptide on the developmental competence of mouse embryos generated through round spermatocyte injection in 2017. This slide shows effects of scriptide on development-related gene expression. Methylation levels of H19 and SNRPL return to the same levels following ICSI. Embryonic development was improved with addition of scriptide in the medium as much as ICSI group. Now you can see the blood cyst rate is 74% from ICSI and 59% from ROSI prescriptide. And the number of third-term offspring percentage is 45% from ICSI and 40% from ROSI with scriptide. You can understand well that the lossy with scriptide group showed significantly higher embryonic development. Lossy result was in a clinic data, and the lossy with scriptide data was experiment. In Japan, scriptide is allowed to use for the experiment, but not allowed for a clinical application. Summary: treatment of lossy mouse zygotes with scriptide increased the expression levels of several development-related genes at the process stage, resulting in more efficient in vitro development of the process and an increased birth rate of lossy-derived embryos. In conclusion, this study reconsiders ROSI and acknowledges its high potential to help many non-obstructive azoospermic men. However, further study to find optimal conditions should be necessary. It's my hope that the benefits of lossy are recognized and that the successful results that I have obtained can be replicated by any doctor anywhere, and that the treatment becomes more widely used around the world. I am willing to put my best effort to help anyone of you who is interested in mastering this treatment. Thank you for your kind attention.